Welcome back, or welcome if this is your first time to Health for All. For those of you who are new to Health for All, this YouTube channel discusses widely debated practices in healthcare and widely debated topics in medicine. I know this may sound negative, but the point of this channel is not to criticize the fields, rather, it's to bring awareness to them. Anyways, it is important to note that the kind of videos made in this channel may change over time as the channel expands. Now, before we delve into today's topic, please consider subscribing if you have not already. If you have subscribed, thank you so much. I truly appreciate any and all support I receive. So please consider subscribing. This video will be covering the controversy around genetic editing. But first, what is genetic editing? Genetic editing, also referred to as simply gene editing or genome editing, are techniques that allow scientists to change the genetic makeup, also known as the DNA, of an organism. These techniques either remove, add, or modify segments of the DNA of an organism in order to ultimately change the genetic makeup of these organisms. Over time, there have been a number of techniques developed, but most are shadowed by CRISPR. CRISPR, unlike older genetic editing techniques, is really efficient. It's fast and accurate for the most part. And it's also quite cheap compared to these older genetic editing techniques. Discuss how genome editing is used today. According to the National Human Genome Research Institute, researchers rely on genome editing tools as a way to explore the connection between genotype and phenotype. Genotype refers to an organism's genetic information, while phenotype refers to an organism's observable traits. I've attached a link in the description if any of you are interested in reading more about genotype and phenotype. Anyways, to elaborate, scientists often study how different genes affect humans by editing, removing, or modifying segments of DNA of mice, zebrafish, and other animals. These animals are chosen because of their genes' similarity to humans. In fact, the National Human Genome Research Institute revealed that mice and humans share about 85% of their genes. That's a lot. It is important to note, however, that genome editing can be done on other organisms as well, not just animals. In fact, I used CRISPR technology on E. coli to make them glow like how glowfish glow. But most importantly, scientists are developing gene therapies. Simply put, gene therapies are methods trying to prevent and treat diseases. As you may be able to tell, being able to use genome editing to prevent and treat diseases would have lasting impacts on our world. It has a lot of potential. There are two kinds of gene therapies: germline therapy, which edits the DNA in reproductive cells and thus passes these new traits across generations, and somatic therapy, which edits DNA in other cells and affects only the organism whose DNA was edited. Apart from treating and preventing diseases, there has been debate about whether genetic editing techniques should be used for non health related purposes, like bettering one's athletic ability. Now, this isn't directly related to humans, but I find it fascinating. Although you may not realize it, you most likely interact with organisms that have edited genes on a daily basis. What I'm referring to are GMOs, aka genetically modified organisms. The term GMOs refers to food products. You most likely have seen this on food packaging. These crops are edited to produce more output with less labor. Not just this, but they also become resistant to certain factors that hinder their growth like pests. An example of this is corn. Most corn in the United States is a GMO, it's gene edited. Now, I just thought that was interesting to share. Let's now discuss the pros and cons of genome editing, specifically in humans, starting with the pros. Clearly, genome editing has a lot of potential to better our world. As mentioned before, it can cure diseases and genetic disorders. Here's an example. In 2015, scientists successfully used somatic gene therapy when a 1-year-old in the United Kingdom named Layla received a gene editing treatment to help her fight leukemia, a type of cancer. These scientists did not use CRISPR to treat Layla, and instead used another genome editing technology called TALENs. Doctors tried many treatments before this, but none of them seemed to work, so scientists received special permission to treat Layla using gene therapy. This therapy saved Layla's life. So clearly, this is really influential and has a lot of potential. Layla's life was saved because of gene editing techniques. She had cancer, leukemia specifically, but because of gene editing techniques, she was able to survive. This is really influential, has a lot of potential. Oh, that rhymes. So this has really great potential. This can do a lot for our world. Similarly, genetic editing techniques have the potential to prevent the spread of hereditary diseases. That is also really influential for our world. Furthermore, gene editing can pave the way for more personalized approaches to medicine, as we can tailor treatments based on an individual's genetic makeup. This leads to more efficiency. In addition, genetic editing techniques have the potential to reduce healthcare costs, as by potentially limiting the need for long term management of genetic diseases and any other diseases, gene editing could reduce the overall cost of healthcare related to these conditions. Also, gene editing could allow us to make organs that match a patient's immune system, which would ultimately help solve the problem of organ shortages and increase the success of transplants. That is really influential. I've been saying that a lot, but all these things are really influential. But ultimately, genome editing allows us to understand genetics in humans and allows us to learn how certain genes affect humans. Now, onto the cons. Gene editing in humans has a lot of concerns that must be addressed, one of them being related to CRISPR. CRISPR is much better than older gene editing techniques. It probably is our best genome editing technique we have currently, but it isn't accurate all of the time. For example, sometimes genome editing tools cut in the wrong spot. Scientists are not yet sure how these errors might affect patients. Assessing the safety of gene therapies and improving upon genome editing technologies are critical steps to ensure that this technology is ready for use in patients. So, should we take that risk that it could go wrong? The editing we do to our genes could go wrong. And also, should we wait a bit more until we have developed more efficient, more accurate genome editing technologies? Or should we go ahead and take that risk? Furthermore, making fundamental changes at the molecular level that can impact an entire life requires careful consideration. There could be other unknown consequences to these changes we make to our genes, and they could be negative. In addition, applying gene therapy to an embryo, given that the embryo cannot provide consent, raises ethical questions, one of them being if parental consent is enough. Since embryos can't give consent that they are fine being edited, we have to ask ourselves, is parent consent enough or should we just not edit embryos? Moreover, if gene therapies become prohibitively expensive, only the wealthy may have access to these techniques, which could worsen the inequalities between the poor and the rich in healthcare, and just in general. Apart from those points, there's also a risk that genome editing can be used for non medical reasons, like, as mentioned before, improving one's athletic ability. Should we allow this? Should we not? Is this appropriate? Furthermore, the authority of scientists to alter germline cells is significant, as these changes will be carried across generations. Should we allow this? Is this okay? Genetic modifications could also threaten the biodiversity that is crucial for evolution. And reducing this diversity might deprive future generations of the diversity that we get to enjoy today. So, at the end of the day, there are many benefits and negatives of genetic editing techniques, and many purposes. The ones listed in this video are the major and most debated ones. I will attach links in the description if you want to do further reading, further exploration, and I'll also attach the articles and readings I used to make this video. Also, just a side note, most of the points for the cons or negatives of gene editing I got from the National Human Genome Research Institute. Just if you're wondering, I'll attach those articles in the description below as well. So now the time has come. What do you think about genetic editing techniques? Do you think they should be allowed? Do you think they shouldn't? Do you think there should be certain restrictions on genome editing? And if you don't know what you think about this, that's okay too. Sometimes you can't choose a side yet, and that's okay. Now, I hope you enjoyed this video. If you did, please consider subscribing and liking the video. I appreciate all the support I can get. These videos take a lot of time to research, plan, record, and edit. In fact, it was 2:10 a.m. when I wrote my first draft of the video. But anyways, please consider subscribing. I really do appreciate all the support I can get so I can further educate people about debates in healthcare and medicine. Thank you so much. Thanks for watching.